So I won't take any of your time.
Super.
There you go.
Thanks, Mike.
And thank you all for being here. I wanted to, Mike keeps mentioning me, but this is all about Taylor. She's going to bring the average age of the presenters down a little bit. I just wanted to, before I introduce Taylor, just mention a little bit that what I find inspiration in as a scientist. And first of all is, of course, all of you in the audience. You know, it's been fantastic being here for the last 6 years. I'm moving to Iowa, I'm moving to Wisconsin in 2 months. So I'll be headed back home. I'll be the associate director of a freshwater center in Ashland, Wisconsin, on the shores of Lake Superior. So I'm excited about that change and transition professionally and personally. But it's been great being here, and I've really been inspired by all of you that have been doing so much. Right? I think of to be a little cheesy because I'm from Wisconsin. You know, the Lorax, the end of the Lorax says, unless someone like you cares a whole awful lot, nothing's going to get better. It's not. And it's been inspiring being here in Iowa and meeting so many of you and seeing all the work you're doing. The Raccoon River Watershed Association, the Iowa Rivers Revival, working with Neil at Drake. It's just fantastic, and things will get better. And if they don't, then we've at least fought hard. So thanks for that inspiration. The second source of my inspiration, you'll see a little bit of that here, but also more in the crowd, are of course the next generation, the students who are learning about these issues and who are going to make really meaningful change. I don't mean to be that generation that looks down at the younger students and says, okay, sorry about all that, how about you fix it? But I do have 2 young children, and I think about that a lot, and that's what's really attracted me to teaching, which I'll continue to do. And I think Taylor's a great example of what the future holds. So thank you to the students and the younger generation for the inspiration as well. So speaking of Taylor, she's from Seneca, Illinois. She's graduating from Drake in about a month. She started her interest in water quality as an 8th grader when she, for the middle school science fair, decided to collect water quality samples in the Illinois River downstream of the nuclear power plant in Seneca, Illinois. So her interest in water quality started ages ago in terms of your youthful age. And what she's going to present on now is some research that she did funded by the Raccoon River Watershed Association. It's really fantastic that you all fund research because you want to know what is out there and what is happening in our streams and rivers. So Taylor, I will say, we wrote this together. We've been working with Dr. Shauna Kaplan as well at Drake. She's a microbiologist, but Taylor did all of the field work, even though my rule is don't go in the field alone. I turned the other way for Taylor. I trusted her. She came back some days super muddy, and we fallen down the banks because it was, you know, Iowa streams and rivers. But she did a really fantastic job with this project. It's been very independent on her end. And what's really exciting, thinking about the inspiration that these students provide, Taylor is going to be continuing her studies on water quality here in Iowa as a graduate student with Michelle Superior at Iowa State University. And she's starting to collect data like June?
The day after graduation.
The day after graduation. Yeah. So she is going right from Drake and our lab on water quality to Michelle's fantastic research group at Iowa State. And I really look forward to seeing what Taylor does, both as a graduate student at Iowa State, but also in her career thereafter. So, Taylor.
Like I don't even have to talk, you guys are talking. And I will point out my dad is a biology teacher and a fishing coach, so that makes the nuclear power plant thing make a little bit more sense there. So yeah, today I'm going to be talking about the recreational risks in the Raccoon River and if E. coli concentrations accurately represent pathogen risk. Again, I want to thank Raccoon River Watershed Association, Polk County Soil Water Conservation District, and as well as Des Moines Waterworks. This has provided me with a great opportunity and has, improved my researching skills in a whole lot of different aspects. So first, I'm going to kind of talk about why we decided to study E. coli, right, as a water quality predictor. But first, I want to take a step back and talk a little bit about ICON. I'm sure most of us are familiar with the Iowa confluence of water trails, but I really want to emphasize how important this is and how important E. coli is to this study. They're going to be incorporating more than 80 installments over 150 miles of streams and river in the central Iowa area. And this is really important. It provides an opportunity for a lot of people to connect and physically connect, with the water sources and the natural capital that we've been talking about all this morning and afternoon. And it really drives an important part to commitment to water safety and conservation in the Iowa area. As somebody else was just talking about, it raises this issue of balancing the safety of water with encouraging people to go recreate in it. How are we going to create systems where families are comfortable bringing their kids into? And how do we balance encouraging people to get out there while also ensuring, their safety and that they're not going to get sick? So I want to just kind of do a brief little overview of E. coli and how I'm going to be talking about it today. So the first part, I'm going to be talking about E. coli as just a general idea, as a general standard, because right now the only EPA standard is for 235 colony forming units per 100 milliliters of water. And that's just talking about general E. coli. It's not specific for any certain strain or any certain gene or anything. It's just this like one big general concept. But this is really crucial to our study because not all strains of E. coli are bad. A lot come from animal sources that quite frankly don't harm humans. And we don't really talk about that enough. However, there are certain strains to be concerned about because if you do ingest them, you can get various sorts of sickness. And also current E. coli samples take about 24 hours to get the results from, if you're lucky, because they all require an 18 to 22 hour incubation period. So if you're wanting to go, it looks sunny one day, you want to take your kids in the water, but it rained overnight. If you take a sample from that day, you're not going to get an accurate representation till at least about 24 hours later. This obviously presents a really clear issue. And there is some work being done in the metro right now to identify real-time metrics, samples that you can see as a clear predictor of E. coli that I'll talk about a little bit later. And on the left there's just one of our sites that I'll be talking about here pretty soon. So today I'm going to be breaking this down into 3 different ideas. The first being how do temporal and spatial influences affect E. coli values, what toxin genes are present, the certain strains that are harmful that I just talked about, then if there are any environmental variables that accurately predict E. coli values. So first I'm going to give you a quick little layout of the sites that we did sample. This is pulled directly from the ICON website because 4 out of the 5 different sites I talked about are going to be, locations out of the 80 plus installments. The 5th one is going to be at Grace Lake, and if you've ever been to Grace Lake, it's a really highly recreated area anyway, so it still did benefit our study. So our first site here is going to be at North Walnut. That's at Walker-Johnston Park, right by the Urbandale Library. I'm going to be talking about all my streams in order of stream size. So North Walnut is going to be our smallest stream, and then we're going to move into Walnut Up, and that's at the Walnut Regional Park. And then followed by Walnut Down, which is going to be right at Colby Park. And then our last 2 are going to be obviously in the biggest stream size, the Raccoon River, the first being at Walnut Wood State Park, and the last finally being at Grace Lake, which is again the one that isn't technically a study, but is really recreated in any way. And I do want to point out that you can see actually on the list right over there that it's kind of small, but all of these sites, they have intended uses of wading, a few fishing, and carrying down sites, which all imply physical contact with the water, which is why these sites are super important here. So I'm going to show you some general E. coli data. Again, just E. coli as a general concept, not any specific strain. I want to point out the size of the streams here that you can see. The yellow is going to be the North Walnut, again, the smallest. Moving into Walnut Down, that's at Colby Park. And then the last being obviously a much larger body of water at Walnut Woods State Park. The data I'm about to show you, the general E. coli values going up, and then I'm going to have it in order from left to right of stream size, starting with the smallest and increasing all the way to Grace Lake here. The red line that I show is the EPA value. These values are averages of the 8 different days that I took samples on. The 5 different days, 5 different sites, 8 days each, for a total of 40 samples here. So you can see a really clear pattern here is that as the stream size increases, the general E. coli decreases. which intuitively makes sense, right? There's more water there, it's going to become more diluted over time as that bacteria and such travels downstream. So these values here are going to be out of a total of 8 different days. And that's the number of days, again, out of 8, that my sample exceeded the EPA value of 235. You can see that the only one that averaged under is at Raccoon River Walnut Woods State Park. Grace Lake was just over it. But yeah, there's a really clear pattern here and a really clear difference, right? 7 out of 8 days at North Walnut exceeded the EPA values or standards, whereas the Walnut Woods State Park only 2 days exceeded. And the only 2 days that did exceed, one value was 300, so just, you know, a mere difference over that 235, and then the last one is 730, which in the big scheme of things isn't anything crazy, which is kind of sad to say, given it's about 3 times the standard, right? Yeah, so now I'm going to be talking about this other study that I've been fortunate to be a part of in Peter's lab. We have E. coli data over a period of 2 years on Walnut Creek. We had 3 different locations. They're not the same as the ones I tested for this, but we have an ag location, suburban, and urban. So yeah, some general trends you can see along here is that E. coli values are typically highest in late summer to early fall. And then this little square here is the time span that I sampled for this study. The first day was August 23rd and the last was October 11th. One really important thing that you want to note, in the big scheme of things, you know, the time I sampled, this wasn't the highest point of time. You can see on the left here, there's a lot of values that are a lot higher, right? But it's also not the lowest either, so we have a good mix. Whereas near the end of October, on the right side of that red box, it is, you know, pretty low. So again, that's the same data I just showed you. And then I'm going to show you coming up here the values for the 8 different days that I sampled. So here we have these. Again, remember that red, as you can see on the bottom there is really low, is the really big locations, both on Raccoon River. Whereas at the top, the different blues are on Walnut, and the yellow is our highest point, North Walnut. I do want to point out this is on a log scale, just for reference. But yeah, there's really clear, you know, patterns that those at the big water systems, the E. coli values were really low, which again makes sense. You can see that most of those yellow dots, again, 7 out of 8 days, were above the standard here. And so some big takeaways that I want to talk about is that E. coli is higher in the smaller streams, right? Which, if I want to tie this back to recreation here, really poses a big issue, because if you're a family, you know, with little kids, where are you going to take your kids to go recreate? You're going to take them in the small streams that looks a lot safer versus at the really big, you know, you're not going to take your kids to swim in Walnut Woods State Park or Grace Lake, most likely, because it's big and deep versus the small and shallow streams. But those small and shallow streams are where E. coli values are highest, which really, again, makes us think about that balance of how do we want to present, you know, this is safe water, but, you know, we want people to be in it, because that really complicates that issue. However, highly variable E. coli demonstrates the difficulty of understanding water quality. You know, there were some trends, however, it's not like there was a really clear pattern or anything, right? And so it really just reminds us of how complicated water quality as a concept truly is. So now I'm going to be moving into what toxin genes are present. So we're going to be moving away from the idea of general E. coli into like the nitty-gritty specific strands or strains. So just a very brief overview of how we got down to this little DNA and RNA, which is quite different than the idea of, you know, big water samples. After I got the sample, I just took it back to the lab, I filtered it, and I worked with Dr. Shauna Kaplan to isolate DNA and RNA, and then we ran some real-time PCR. I won't go into the details now, but if you have any questions, I'm more than happy to do my best to explain that to you. So this chart here shows all the different strains we did look at. There's an asterisk by the last 3, being rotavirus, giardia, and human bacteroides. When you're running PCR, you have to have a certain gene to amplify the DNA that's in a water sample. And sometimes those genes don't always work. So I guess that's more exciting that I'll have more data to come, but I don't have any for those 3 right now. And then I have 2 asterisks by rotavirus because that's the only RNA strand. Everything else comes from DNA. So I do want to point out that we have data for both Shiga toxin 1 and 2, referenced as SGX 1 and 2. This is a really, commonly known harmful strain of E. coli. So I'm really excited to have that data. And then I also have EAE, because I don't want to pronounce that big long thing. And then also Salmonella as well. A lot of these, they come from human and animal feces, as well as Shiga toxin 1 and 2 can come from contaminated food and raw meat. And you might ask yourself, why is this important with water? Why is there raw meat getting in our water, right? But it's important to think about, both urban and agricultural influences, that being, agriculture runoff, as I'm sure we're all familiar with, but then again, leaky sewer systems as well. Both can contribute to this issue of E. coli in water in different ways. And then again, various symptoms, I won't gross you out with that right now, but just know that they can all get you, pretty sick in one way or another. So now I'm going to show you data across showing, potential pathogens are present. But I want to add a little caveat here. So when I'm about to show you the data, it's going to say POS for positive, but I want you to know that it's going to be potentially positive. So without getting into, the very fine details of running PCR here, what you need to know is that when you're looking for a certain gene, you're going to have two wells. We ran this data in duplicate. And so what happened with these potentially positives is that one came up saying, yeah, you have the gene here, you have the DNA. But the other one was like, there's nothing here. So what we're going to do to fix this issue is that we're going to run it again, but in triplicate, and hope that that gives us, a better idea. And so on this graph here, I have the dates, 8 different dates going across, and 5 going to 5 different locations going down below. So if we're looking just across, that's going to tell us temporally patterns, and if we're looking down, we're going to see spatial patterns. So going across those 4 different dates, averaging around 3 to 4 typically, are out of a total of 8 different dates that I sampled. You'll see a parenthesis number after a few of these. That's because we sampled for 4 different genes, right? And so that's out of a total of 4. You can see that there's only a few where 2 out of the 4 strains came up positive, but the rest, there's typically only 1 or whatnot. And then if we're looking down, you'll see that's by date across all 5 different sites. So that's going to be out of a total of 5. There is a little bit more variance here, given that there's 3 different days where only 1 came back positive. Whereas if you look at August 30th, that's going to be the second column for you. 4 out of the 5 days came back with something, and one of those had 3 out of the 4 different strains positive. So, that implies, again, this is potentially positive, so you can't say anything for certain, but that definitely suggests that there's an issue, to say the least, with the water there. So again, some main ideas I want you to get from that is that there are potential positive across all different dates and all different sites. There is some variation here, but again, we're going to have more RNA and more DNA tests to run, so that's really exciting, and to hopefully get some confirmation on those things there. So now I'm going to kind of shift into looking at environmental variables and to see if, is there a real-time metric that we can get that predicts E. coli accurately. So first, before I do that, I want to define a few terms for you. The first being turbidity, the quality of being cloudy or thick with suspended matter. If you think of chocolate milk as water, that'd be a really high turbidity value. And then also dry mass, a very similar in concept, it being the measure of suspended sediment. And these are really important to tie in with E. coli values, specifically because rain events flush water systems, as we all know. If you have a small stream, you have a heavy rain event, the stream bank is going to fall in because of erosion, and you're going to have a lot of sediment throughout the water, leading to high turbidity and high dry mass values as well. I'm really proud of these pictures because I didn't know I was going to be doing this presentation my freshman year. But I have pictures from the same site along Walnut Creek throughout the time periods. The second one you can see there, it looks like chocolate milk, right? And that's actually Peter there in the stream, who's not exactly a short guy. So that just shows you how high the water levels were, whereas the other two, the top and bottom ones, the water looks pretty clear. If it was warm enough, you might take your kids to go recreate in there because it does happen to look safe. So now I'm going to show you some data for dry mass and turbidity patterns, the first being dry mass here on the left for the 8 different sites going across. As you can see, it kind of looks like a mess, right? It's very highly variable, but there are a few patterns here, whether that's good or bad. The bottom, that navy blue there, again, the 8 lowest, that's from Colby Park, so that's near a Walnut Down site, obviously average to lowest, whereas the blue site, the bright blue site at the very top, is also from Walnut, just a Walnut up. So this really goes to show how highly variable these systems can be and how hard it can be to predict things like this. And then on the right here, we have turbidity. Again, really highly variable, typically low. However, it is, it's a generally straight line. So maybe this suggests that it is like predictable at low levels for turbidity. And so now I'm going to have values here with dry mass and turbidity, but using them to predict E. coli. So the first on the left is going to be your dry mass. Again, you can see that's the same data. It's a really low R-squared value here, right? It's 0.1505, which is, how much of your dry mass what percentage can you use to predict E. coli? So that's right around 15%, which really isn't that great. And then same for turbidity, except the R-squared value is even lower. It's 0.0555. However, there is a study being done right now at University of Iowa that suggests this threshold right here, right at 20 NTUs. And they say that, if you have this level of turbidity right at 20, that your E. coli value is most likely over that 235 EPA standard, which you can see everything to the left is, it's pretty high, but most of our values were also in that range. So again, our main point from that we want to take away is turbidity and dry mass are poor predictors for our study. It might work for others, but for ours, it didn't work that well. So now kind of breaking it down to the big ideas that we looked at. These are the same pictures we just looked at however long ago. The left being the smallest stream, the biggest on the right. And I want to bring it back to that question of, where would you feel comfortable bringing your kids to recreate? Visually, you're going to bring them to North Walnut or, that Walnut Down site with the little gravel area because it looks a lot safer, right? Versus the Raccoon River. However, our data suggests that, maybe this isn't the best idea. And how do we implement, all these icon sites when we know that, maybe these small, safe-looking sites aren't the best when you break it down to the bacterial level of things. And then this also suggests that, things are highly variable. All this data is highly variable, and it really makes you realize the importance of your sampling frequency, which is what I think is going to be really important when we do implement these various sites. And also that it raises the importance of we need to focus on not only where we're recreating, but when. This is the same exact site. Obviously, you can see the height difference of the water. It reminds us that there's a lot more issues that we need to think about, even outside of bacteria and outside of things like E. coli, but we need to think about discharge as well. Kids drowning. I can tell you, I did not put a single foot in that stream on that day because it was going so fast and it was so high. It probably would have gone up past my waders. But yeah, and it really makes us think about, if we have to worry about this E. coli and this bacteria, but we also have to worry about, how are these rain events affecting the water and where would you bring your kid to recreate? Yeah, so again, I just want to thank Raccoon River Watershed Association and the Polk County Water Conservation District for funding, as long as we got help with from Claire Ruby, and then Alyssa, who's here from the lab, and Morgan, another student there. And I'm happy to answer any questions if I have time as well.
You do have time.
I do have time. I'll do my best.
If I were to draw a conclusion here, I'd say that the pathogenic threat is greatest at low flow conditions, just as sort of a general conclusion. So, to draw a bigger picture, you don't have to measure turbidity or anything like that. Low flow, high concentration, high flow, dilution, and low concentration.
Right.
Is that kind of a ballpark free to summarize it?
Yeah, I mean, and I have some just in case figures here. So I'll pull one of those up because I have it anyway. And so yeah, what you're talking about with discharge really ties into rain events here. And so it's kind of interesting to tie it back during that I can go all the way back. But on the 27th of September, we had typically higher than normal E. coli values. And that's because I was sampling while it was raining. We tried to pick days where, where people are going to be out and about, but I can't predict the weather. So it started pouring on me. But yeah, obviously, you're going to have higher E. coli values then. And that makes sense completely with what you're talking about. Hopefully that.
Can you switch back to that last screen just for a second?
Yeah.
No, the next one.
Next one.
No, that one. I
This
think
one?
just for a second,
Yeah.
because somebody wanted to see it and I had it on you.
Do they have any questions? This is kind of my just in case slide for PCR, if anybody asks me about it. So, yeah.
So if you have family come to you and say, when can my child play in the creek? What should I look for? What should I do? Is there anything they can do?
You don't go when it looks like chocolate milk, but you want to bring your kid there anyway, right? Right now, I mean, there's really no way to know because it's so highly variable. And I think that's something that, obviously it's been a question on all of our minds, but this really emphasizes how much we don't know about things and how much more research, needs to be done in this area.
Or maybe go after a rain event when you think everything's been kind of washed out?
No.
Well, that's when it is all over.
That's not what we found, that's not what they found.
Kind of the opposite.
The opposite.
Yeah, so if you have,
Do not go into the
yeah, so if
water.
you have a rain event,
You have a big rain event.
one day that happened, everything's flushing in the system. All the things from the stream bank are now in the water that weren't there before. Whereas if it hasn't been rained and everything's kind of sitting, you're not going to have the inputs from the land, from if there's a leaky sewer system nearby, or if there's a farm nearby with animal waste or whatnot, if that makes sense. Yeah.
What do you think is the, I guess it's with the pulp mill, what do you think is the biggest sort of relative risk of the water? So you talked about bacteria, we also have the microtoxins and so forth. Do you have a sense of what we should really be paying the most attention to? Because we only have limited resources.
Yeah, that's a loaded question. Well, I think sugar toxin 1 and 2, I know there's specific strains of E. coli, but I mean, that's a really well-studied, strain of DNA that, it's known to be waterborne and it's known to be really harmful. So I think these things really are important, especially because, if you take your kids to the water, who's most likely to accidentally ingest water? It's not going to be the adults, it's going to be the little kids who you want in the small streams. And I think that's why this, and dogs too, right? And I think that's why this is really important. I know that didn't really answer your question, but I don't know how to pick one specific thing.
I'll offer a
Yes,
follow-up. and
please do.
In Michelle Zucker's lab,
Okay,
you're definitely going to be hearing about.
I'm
and
excited.
Perhaps working with Michelle, it's her lab who did our local samples
Oh, okay.
on the ISU campus. But I would posit that antibiotic resistant genes, as well as the whole bacteria that she's been talking about. And so right now I'm working on a term paper for a diagnostic microbiology class that is focused on something called the KPC gene. But it's that bacteria that it's mostly first found in is a close relative of E. coli. So you all who have been involved in water quality monitoring or reading the paper know that we're talking about E. coli as a fecal indicator bacteria, and it's, as she's proven to us, it's not necessarily the toxic strains of those bacteria. But the thing with the gene that I picked is that it moves around to E. coli and like at least 10 other species in that particular family of bacteria. So those are coming out of us, particularly if we're in the hospital, where
Mhm.
they're nuking us with antibiotics to try to often save our lives.
Mhm.
And
Public health.
so hospital wastewater
Yeah.
is one
CAFOs.
contribute to that, but basically it's happening everywhere. The problem with those genes and types of resistant bacteria is what's happening is they're now strains for which no known antibiotic could stop it in your body or in your lifestyle. So that's happening. So that would be my argument that we can see that.
Yeah, I think I saw you raise your hand a bit ago.
I have, Claire Ruby just has a question. She would like to ask, what are some of the hurdles we're getting the lab methods up and running? What were some of the hurdles?
So our cyber green is what we used, is the gene that we used for all the data that we do have. So that worked really well. But apparently the specific primers that she was using for all the other ones, it's all isolated and everything right now and just waiting. But yeah, I think it was the primers. Hopefully that answers your question for the specifics. Yeah.
We want to give the floor to Tom, but I just wanted to make one observation that Taylor and I also discussed. The day that was hottest in terms of potentially positive pathogens was the 30th. And you can see generally coli measurements, 3 or 4 of the 5 sites were under the threshold of EPA, suggesting that you could go in the water and even if EPA said, oh yeah, it's fine, that's when we detected the most number of potentially positives. So that's another downside to that method. But
And I'm happy to answer questions after.
yeah. Yeah.
Did you spend a lot of time on the water? Thank you very much. We're gonna have you.